Okay, so let's get restarted. So we here when I was trying to explain the concept of the receptive field, and the first part of the brain that encounters this concept is already at the level of the retina here. So as I explained last week, the light goes from the bottom here like this, and then here's the fovea, the highest resolution of the eye, and this way the light is coming, and then it turns out that cones and the rods are located at the back of the retina. So here, around here, there is some kind of light scattering and things happening, but eventually it gets to this part. And then these cones and rods generate some kind of electrical activity, electrochemical activity that is passed on to by different kind of cells called bipolar cells and amacrine cells and so on, which I'll skip. And then eventually come to this retinal ganglion cell, and then that passes output into the brain through thalamus. Okay, and then if you put the electrode into here, so-called optic nerve, then you can see some kind of pulse going on once in a while. This vertical thing, this is about across a time, means that there is a sort of a brief impulse of activity that is detected by here. And then if you place another different electrode into this retinal ganglion cells, then you see also the same kind of activity here. But that's what we mean by the first activity of the neurons in the retinal ganglion cells and also optic nerves. By the way, then here I wanted to ask you a question. So retinal ganglion cell is already a neuron, okay? So when there's a neuron like this way, and then this one has a really long output. What's the name of this part, which collects the input from other neuron? Can anybody say in the chat? If not, then there. Yeah, okay. So that's a question. So the output. So Suleiman is actually, in a sense, correct that it's either dendrites or axons, and the output is also either dendrites or axons, right? So then the question, Paul, here is that which one is dendrite and which one is the axon? Option A is that the input is the dendrite, and then option B is the input is the axon. Which one is your option? Poll, and then relaunch. Okay, so that was already quick in the answer, I guess. So, all right. So yes, the correct answer is this A. Input is called dendrite. Okay, and this is something that I am likely to ask also in the quiz. So make sure that you understand it. If everybody already knows it, then I don't need to ask. All right, so then let's go to the next one. The input is a dendrite. And then this is two types of the retinal ganglion cells. One is having a receptive field that corresponds to somewhere around at the center of this visual field. So when the neurons so the central part of the activity, central part of the responsibility falls around here. And then if you place light spot like this way, this on-center cell becomes very active when there's something bright at the location of the receptive field, like here. But it becomes completely suppressed when bright thing is around and then dark thing is the center. And then intermediate when it's uniformly black or uniformly white. And the off-center cell is basically doing the opposite. So if it's uniformly black or uniformly white, it's intermediate. But if it's spot of white one or spot of the black one, this one, spot of the black one, makes a huge response. And it also is important to know that this is also the case at the time of the turning off the stimulus. When you turn off this non-responsive stimuli, then it induces a lot of response here. Same here, lots of response here. But if you remove the stimulus that evokes the activity from the cell, that suppresses the activity here, suppresses activity here. And that is important to consider after image later. Okay, so here's the map or how it looks like in terms of the dendrites. If you image it at the level of the retina. So there are two types of the cells in the retina, but it's not super important for us at the moment. But small types of the cell called the midget cells, or bigger types of the cells called the parasol cells in the retina. And then you see three types of the size of the cells, right? So these are both retinal ganglion cells, and they become very small to medium to really large. And then why is it? It's because their size increases steadily when moving away from the fovea. So remember that when the eyes are like this, and then when the fovea is here, then the size of the ganglion cells is here very small. But as you go into intermediate, then it starts to become bigger. And then if you go to the periphery, it becomes really big. So even if the density of the cone is very, very, tight here and then sparse here, and even sparser here, because this guy can catch a lot, it can be sensitive to the color sensitive cells, color sensitive input. That's a sort of the intuition that you want to get from here. Okay. If you don't have a question, then I'll just go further. So the size of the retinal ganglion cell and the extent of the dendrites determine the extent of the receptive field, because it is size or the extent of the dendrites are the determinants of how this cell responds towards input from the outside. So wider dendrite collects inputs from retinal ganglion cells across a wider area. And schematically speaking, the upper part is the retinal ganglion cells, and going from small rods and cones here. If you are in the periphery, you can collect the input from a wider area to make a response and then send it to brain. So that can potentially explain why our phenomenology in the periphery is colorful, despite the density of the cones are sparse over there. So dendrites are wide at the periphery, a common property across layers of visual processing. So here, when you are seeing this type of the stimulus, okay, and then at the level of the retina and also the fovea, you see, small neurons, retinal ganglion cells are receiving the input, and then it can resolve these different kind of color, and that is sent to thalamus LGN to B1. And the B1 also collects the neurons that has because this one has a slightly bigger receptive dendrites compared to LGN or retina, then it can combine input from 3 LGN cells to make it active like this, or sensitive to the color. And at the periphery, retinal ganglion cells itself can collect the wider areas of the brain, the visual input here. And then that can further collect, can be collected within the V1, because it has also correspondingly wider dendrites field. Okay, so the sum up here, the neurons that respond to the peripheral visual inputs reflect inputs from the cones over wider areas than those that respond to the foveal visual inputs. And therefore, the convergence effect gets stronger. And that effect starts to become stronger for the neurons at the higher visual hierarchy compared to retina. V1 has such a property, and V1 is even superseded by other neurons in the higher visual areas that I'm going to talk about in a couple of minutes. And assuming that color experiences arise from the higher areas, big and uniform surface should evoke stronger color experience. That's the prediction from this type of the structure of the visual field. Is there any questions so far? That's fine and clear so far? Okay. What was the convergence effect? So the convergence effect means, yeah, maybe I should have explained it better. So because V1 neuron here also has a big, receptor dendrites compared to the retinal neurons like here. V1 receives an input from the retinal ganglion cells that covers this area with these dendrites, for example. And then this retinal ganglion cell, which covers this part of the visual field, also is covered by here. And these are converging to this neuron. And that's what I mean by convergence effect. Is that clear? Okay, all right. So let's move on. If there's any other further questions, if not, I'll move on. On the other hand, now it probably is much clearer for you about if the stimulus are not scaled, towards the periphery, what happens is the following. So see here on the left side, input is now uniformly sized. Okay, so something at the center, there is a red patch, but very, very small. And then these are all stimulated. These, small red stimulus can stimulate, group of retinal ganglion cells within the fovea, and that gives you strong activity in the thalamus LGN, and then converging to V1. And so, this receives an input of very tiny but clear, redness kind of information. On the other hand, if you look at the periphery, so here's what's already happening. Because this retinal ganglion cell collects the input from this area, and each of the input is inconsistent, it starts to respond as if it is responding to broadly, this grayish or bluish kind of stimulus. And the same goes for this part of the retinal ganglion cells. It's almost the same response to that blurry pinkish kind of thing, and also here it's a blurry yellowish thing. And this is all schematic, but the point here is that if the input within the receptor field is not consistent across all the inputs, then the resulting effect becomes blurry or weaker. Remember that on-center or off-center cells responds the highest when there is the most preferred stimulus within their receptor field and anti-stimulus in the surrounding area. So something like this kind of situation is not the best way to evoke response to the neurons in the retina. And then because of the convergence effect, V1 neurons further combines this, blurry kind of input into something really even more blurry. And that continues on and on and on across different visual hierarchy. And therefore, as a result, when the stimulus is not appropriately increased, then you will also lose or you start to see things blurry. And that's what you see when the stimulus is uniformly sized and at the periphery. So just to summarize up to here, if the color stimulus are still small, fixed size in size and random, then neurons at the fovea have a better spatial resolution, but collecting inconsistent color information at the periphery predicts more blurred responses of the neurons responsible for color experience at the periphery. But this is not the same, as we cannot see any color at the periphery. That has been claimed by some philosophers and also prominent psychologists. And also probably you will see this in the psychology textbook, but some of them needs to be updated. Okay, so for any question? If not, then I'll go. So the point here up to now is that don't be fooled by the first impression of this figure. This is quite important. Y-axis is not the number of the cells, but it's the density, okay? Here, it's number of the receptors per square millimeter. So what it means is that to obtain the number of the receptors, you need to multiply by the area that matters. So if you have a very low cone density here at, let's say, minus 60 degree, periphery, then we need to multiply by the area that is covered by one neuron to estimate how many cones that receptor at that location would receive. And as long as it's not zero, then it can become really substantial number. So density needs to be multiplied by the size of the size to obtain the estimate of the number of cones that contributes to the process over there. So in summary, retinal ganglion cells send visual inputs via spikes through LGM to V1, and the size of the receptive field of retinal ganglion cell becomes larger at the periphery. And the same is true for all visual areas. And this can explain why we can see colors of big dots at the periphery, but not small ones. Okay, is there any questions so far? Otherwise, all right. So I think we have 7 more minutes until the next break, so I'll go. So then what are the hierarchical structures of the visual system? That's the next topic. So visual hierarchy can be defined in many ways. And from the retina, visual information is relayed to the brain. The primary visual cortex is the one that we talked right now. And as the one goes higher in the hierarchy, receptor field becomes larger and also more complex. And from V1, hierarchy can be defined based on the anatomical connection rule. And the key anatomical features is cortical layers and also columns that I'm going to explain. So the first component of this anatomical structure related to hierarchy is vertical hierarchy within each area. So the right side figure shows the structure of the neural tissues in each of the different cortical areas. So layer, it's called a 6-layered structure, okay? And it comes with a layer 1, 2, 3, and also 4, and then 5, and then 6. And depending on which area you are in, the number of the neurons and also the thickness of each layer differs. For example, this is the areas of the V1, which receives the input from other lower areas. And these so-called primary sensory areas tend to have a bigger and thicker layer 4. That's an input layer. And then the output layer is this layer 5 that produces some kind of output into other areas. And this tends to be thicker for other types of the areas. Do you have any ideas of the areas that would have a thick layer 5 or layer 6 that also has a output? In chat box, do you have any ideas? What types of the structure should have layer 5 or layer 6 that has a large neurons? Maybe it's a bit difficult. So the answer is motor cortex. Because you want to produce a strong outputs and so many outputs, so-called primary motor area like M1 has a huge layer 5 and with the very diminished layer 4 or even absent. Okay, so any cortical area is composed of 5 to 6 layers in a vertical dimension within a column. And within a given columns, neurons tend to have a similar receptive field size, receptive field properties in terms of location and also size as well, property as well, as I explain later. But here, today, what I want to pay attention is actually so-called connectivity between the area through the layer structure. So, okay, so if one area is lower than other area, okay, so this is the higher area. This is the higher, and then this is lower. Then the pattern of the connection is like from the upper layer into this middle layer, layer 4, or upper and bottom layer to layer 4. For some reason, we don't know, but this is almost always the case in the mammalian brain. And then something similar also happens when you go from lateral away, meaning that, within the same level of the hierarchy, then all the layers of the neurons project into either the upper or lower layer neurons project to everywhere in the hierarchy of the column. And then when you are producing some kind of descending or feedback input, then it tends to produce the input from the lower area to the higher, lower area receives input from higher areas at lower or upper layer. Okay, it's a bit complicated, but in an upshot, let's say if you have two areas of the brain, let's say V2 and then V1. Then V1 is a lower area than the V2. So if you look at the neurons in V1, then you can find neurons in layer 1 or layer 5 or 6 that goes into layer 4 of the V2 because V1 is lower than V2. On the other hand, if you are looking at the V2 to V1 connection, then the neurons in V2 in the higher area, upper layer, layer 1 to 3, or 5 or 6, can project into V1 as a feedback input. And that terminates at the 5, 6, or layer 1 or layer 2. And so this is the kind of anatomical rule of the visual hierarchy. We don't know exactly why this has to be the case, but that's the sort of the empirical finding. And based on this rule, when you apply this rule to all the combination of the connected visual areas, then you can construct this kind of amazing map, or almost like, London's, subway map like thing. And then here you can start to see the layer hierarchical structure of the visual system. And what we have seen is just, retina, and the LGN is a part of the thalamus, and the B1 is just here. So B1 is the entry point into the brain, but the rest of the brain areas of the, related to visual processing is just homogeneous, and there is some kind of, hierarchy here. So up to as you go here, this is a higher area, and then this is a lower area. And then almost all the connections made by this, line is bidirectional. So it means that the V1 to V2, there is a line here. That means that V1 to V2 has this feed-forward connection, terminating a layer 4 of the V2, and V2 has a feedback connection to V1. And the same goes for V1 to V3, and also V1 to V4, and so on. And then V4 to IT, blah, blah, blah. So many kind of complicated anatomical connection arises. So from here on, the anatomical and so neuroscientific study of the vision starts to become really, really complicated. Okay. Okay. So before I'll make a break, I think this is okay. Yeah, I'll stop here, and then we'll take a question after the break.